this biomarker usually is for prognosis prediction, and it's not a screening basis. So we want to develop the new biomarkers for that. And so for the sampling, we go to the literature. The most reliable one is the liquid-based cytology. And the sampling technique, they use this brush-like device, and after swabbing and after liquid-based spin down, and then this slide will be processed under microscope, observed, and then quantified. So in our study, we want to develop a different approach. We want to combine the traditional cytology-based technique and combine it with genetic-based biomarkers on DNA. And of course, it can be measured, it can quantify, so it can exclude the bias, the diagnostic bias. And during the select on some DNA loci, and usually it's correlated with some tumor suppressor gene. And this highly methylation biomarker usually can be detected in almost every type of cancer. So in our study, in a previous large-scale screening on squamous cell carcinoma, we identified two potential high methylated genes. It's PAX1 and ZNF82. And for the training set, the training study, we used our and through our analysis, we actually reach a very high sensitivity and also specificity, both of this gene. And for after the methylation the training study, we want to use a prospective way to see whether this can be detected in another cohort. So we, in this part, we developed another kind of device. It's a cotton roll-like device. And this device can be developed into future clinical use in a non-invasive way. So we also process through the same certification, and you can see we can validate very well of the methylation label, high methylation label, and it's with statistical significance. And in this part, we also collect some cancerous tissue, and from the result, you can see that it seems like the methylation will increase significantly only when the tissue becomes a real cancerous tissue here and here. And you can see here, in the validation state, we also can achieve a very high sensitivity and specificity. And then from the odds ratio, you can see the DNF seems to predict the threshold, the cutoff value, the threshold. You can differentiate between poor prognosis and well prognosis. And the poor prognosis means that the patient developed a metastatic or secondary cancer or recurrence disease after the initial treatment. So some of the biomarkers actually play an important role in the carcinogenesis process. So our biomarker seems to categorize into the tumor suppressor. And then the tumor suppressor gene usually decreased in the cancer, and it's all difficult to detect. But if we use the methylation marker, because it increased, so it became very sensitive to detect these two genes. And we search the literature. Actually, it's not well studied, but we can see here is that both of them seems to play a transcription factor, and if it is a transcription factor and play an important role in the oral carcinogenesis, we will continue to study this, and it's under investigation. So, thank you for this.
Also offering promising results for the clinical oriented activity. I have some experience we have published with the Hopkins University about hypermethylation of the DNA, and I think that is promising. But concerning, you have mentioned sensitivity is high, but for diagnosis, a sensitivity to have biopsy is 100% normally if the pathology is well. So I think that this could be of interest for very early stages. Have you differentiated between both types?
Actually, we do that, but the result is non-significant. It means that no matter the staging, after the lesion becomes a tumor, it's getting high, but there is no difference between the staging. No.
Another situation which is really challenging for us is the second primary tumors and this type of field cancerization that sometimes we have treated the patient and during the control have doubts if the mucosa is changing, and so the be of interest is for this type of patient.
Yes. We have detected that away from the tumor tissue, we also detect a relative higher level of the methylation level on these two genes of these non-cancerous matched tissue. So if we go to the hypothesis of field cancerization, the matched tissue, of course, they are not the tumor tissue yet, but it predict the future development of the second primary or first primary. Yeah.
Thank you, very interesting. So, thank you.
Thank you.